This is a brief explanation of the relevant parts of the brain that are involved in post-traumatic stress disorder. I'm going to discuss this both on the macro level, what happens in the broad circuits of our brain, and at the micro level, that is, what happens at the level of cells and synapses, and share how this perspective informs our understanding of PTSD and the treatment. Now, the first thing to think about is what happens in the brain following a traumatic event. One key process is classical conditioning. That is the pairing of environmental cues with the fearful response that occurred. In general, this is a useful process. It helps us recognize and avoid dangerous situations in the future. For example, if you were walking in the woods and came upon a rattlesnake, it would be good to remember and be appropriately afraid of cues that predicted similar dangers in the future. However, in PTSD, there can be an overgeneralization of this response. Certain cues can trigger a fear reaction, even when the situation is not really life-threatening. You might not even notice the cues at the time of the trauma. They could be smells, or the way the light looks, or even noises that our brain actually notices and encodes, even though it's not in our conscious awareness. Now, because these conditioned cues become paired with the fear response that occurred at the time of the trauma, they trigger a state of physiological hyperarousal that's inappropriate to the situation at hand. The brain does not distinguish past from present. So what parts of the brain are responsible for this process? The amygdala, the prefrontal cortex, the hippocampus, and the anterior cingulate are all important areas involved in fear conditioning and PTSD. Now, the amygdala plays a major role in conditioned fear, associative learning, and the fear response. The lateral nucleus of the amygdala is where the association between conditioned stimuli and unconditioned stimuli get encoded. This process is thought of as fear acquisition. The lateral nucleus then sends signals through the basal nucleus and on to the central amygdala. And the central amygdala is responsible for triggering the fear response via projections to the hypothalamus and the autonomic nervous system. As part of the fear response, the prefrontal cortex gets inhibited. It triggers a reflexive reaction and tells the brain there's no time to think or make a calculated judgment about how to respond. And normally this is protective at times of great danger. Now, normally projections from the hippocampus, memory, the prefrontal cortex, the judgment area, and the anterior cingulate to the amygdala serve as top-down control in regulating and modulating fear response. In individuals exposed to trauma who don't develop PTSD, these areas are able to modulate and dampen the amygdala and the amygdala's response following reminders of the traumatic event. It's important to note these individuals continue to remember the trauma. Their episodic memory is intact, but their prefrontal cortex and other regions help them to understand they're safe. So to treat PTSD, we need to address this type of irrational, conditioned fear response developing in the context of non-threatening cues. The major way we do this is through exposure therapy, and the core of exposure is to write or record a detailed narrative about the trauma. The more vivid sensory details, the more well-elaborated the memory that we can include, the better. And this is then reviewed on a daily basis. It's important to emphasize how challenging this exercise can be. In many ways, it's like experiencing the trauma all over again through the fear conditioning process we've already described. The amygdala is likely then to recognize the conditioned cues and signal a fearful response akin to what happened during the original trauma. However, the prefrontal cortex, hippocampus, and anterior cingulate help to re-regulate the amygdala. And the fear response, every time the narrative is reread, the brain learns, while still it's fear-inducing, these cues are no longer associated with danger. It's teaching the brain that things are not in the present and really not currently dangerous. The brain learns something new. So that's really the macro level of what's happening. Then there's the micro level. In order for conditioning or extinctions to happen, brains have to change. That's really what therapy does in the first place. It stimulates learning and synaptic plasticity and the changing of neural pathways. Synaptic plasticity, or the strengthening or weakening of connections between neurons over time, is really what's central to learning and memory. So let's look at what happens there. There's several underlying mechanisms that cooperate to achieve synaptic plasticity, but one key principle is that connections between neurons are strengthened when neurons fire at the same time. The glutamatergic NMDA receptor plays a key role in this process. This ion channel protein has unique properties that make it ideally suited to learning. Most notably, the channel is blocked when the cell's at rest and only opens when the cell is slightly depolarized. Thus, this NMDA receptor can serve as a coincidence detector. It's a biological signal that's activated when two neurons fire at the same time. And when the channel's activated, the neuron fires and neuroreceptors are increased at the cell surface, thus priming it for future activation. Of course, this receptor also plays a critical role in unlearning conditioned associations, including through the process of extinction. One fascinating finding that's come out of this literature is the idea that NMDA modulating agents could play a key role in the treatment for anxiety. For example, treating patients with D-cycloserine, a chemical that is a partial NMDA agonist, has been shown to enhance exposure therapy in the treatment of acrophobia, fear of heights, by helping it to work more quickly. Now, D-cycloserine has no therapeutic effect by itself. It's only helpful when given in conjunction with the exposure therapy, which is to say, it's not treating anxiety per se. It's enhancing the brain's ability to learn something new. In fact, enhancing synaptic plasticity may be the way that other medicines work in treating PTSD as well. For example, SSRIs, which are often used to treat PTSD, increased brain-derived neurotrophic factor, or BDNF, in the hippocampus, which is important in maintaining the health of neurons and in neuroplasticity. A similar mechanism is thought to be relevant in treating depression, which of course is highly comorbid with PTSD. People who develop PTSD might have a genetic predisposition to difficulties with protein synthesis of BDNF. So as you can see, we can understand the neurobiology of PTSD at a macro level, a circuit that involves the amygdala, prefrontal cortex, anterior cingulate, and hippocampus, and the micro level, thinking about the context of neural firing and synaptic plasticity. Each of these frameworks offers a perspective on current and potential future treatment options. Many of our current treatments, from exposure therapy to basic behavioral interventions, can be conceptualized as helping restore top-down control over limbic activation. Ultimately, interventional approaches may also play a role in restoring circuit balance. Medications that can target synaptic plasticity and learning may also play a critical role in augmenting our therapeutic approaches and helping folks with PTSD.